whenever you come across brain structures, then you can kind of see where they fit in the scheme of things. And this, I kind of came up with this little schematic based on the development, from the ectoderm to the neural tube, and then you had the three outpocketings. And we're going to show those outpocketings there. You had the forebrain, you had the mesencephalon. The forebrain is the proencephalon, and you had the mesencephalon, and you had the rhombencephalon. Those are the three initial outpocketings of the brain. And then they develop into all the different other brain structures. So you can basically divide the brain up into the forebrain, again, which is the proencephalon. And before I go too far with this, I just want to show you, it looks like big words and stuff, but really it's nothing. You have pro, and you have encephalon for all the different names that we're going to be talking about. You'll see this encephalon, and encephalon just means brain. So the only thing that really changes are these prefixes. So like pro is just like the forebrain. That's what it means. And then that's going to be subdivided into two parts. It's going to be subdivided into the telencephalon. And the telencephalon is essentially like your cerebral hemispheres. Telen is end, like communication, like so the end point of like a communication. So it's going to be like end brain. And again, that's going to be like the cerebral hemispheres. And you have right and left cerebral hemispheres. And you can subdivide the cerebral hemispheres. You can break that down into the outer cortex, and you have the middle section, which is the centrum semi-ovale. Then you have the basal ganglion, which is like the innermost gray matter of the brain. Okay, so cerebral hemispheres, but so we're not going to go into all that deep. We're kind of getting you used to some of this stuff for when you go into CNS next quarter. Then we have the diencephalon. So the diencephalon, what does di mean? Two. So it could be two, or it could be like inner, depending upon, your word origin source. It's like your second brain, and it's going to consist of these thalamic structures, the epithalamus, the thalamus, and the hypothalamus. So we'll say second brain, second, or inner brain. Okay, and the diencephalon, again, is going to consist of those thalamic structures. So we're going to have epithalamus. You're going to have the thalamus, and then the hypothalamus. Okay. And I'd like to, and I'll show this like on Friday when we do lecture too, I'd like to kind of associate this diencephalon with the inner brain with the Egyptian symbol for the eye. The hieroglyphic symbol of the eye kind of resembles these things. And you'll see this when we, when we do our lecture part there. I guess I could kind of show you a little bit. So you have like this kind of hieroglyphic type of a look. Get the idea of it. But so, like this part would be like the thalamus. You're going to see that there's an interthalamic adhesion that connects the right and left thalamus going through the third ventricle. And this is like the hypothalamus coming down, and this could possibly represent the pituitary gland. And in the back part, the epithalamus basically would give rise to the pineal gland, which we'll talk about more so when you guys get into endocrinology with me next quarter. But epithalamus, so it's going to give rise to the pineal gland. The pineal gland secretes melatonin, which induces sleep. Actually, there's supplements out there. I just seen it advertised the other day on TV about using melatonin to help you to sleep at nighttime. Then the thalamus, an important thing to remember about the thalamus is that it is the gateway to cerebral cortex. Okay? Gateway to cerebral cortex. So all sensory information from all parts of your body must first go to the thalamus before it's redirected to other parts of your brain for processing. That's why it's called the gateway to the cerebral cortex. And that's true for any kind of sensory information. The only one that isn't, because there's always an exception to something, is the olfactory pathway. And the olfactory pathway has indirect connection with the thalamus. But all other pathways coming in have to go to the thalamus first before they're distributed to other parts of the brain for processing. And then the hypothalamus is going to give rise to the pituitary gland. Okay, the pituitary gland, you got the dental hypophysis, the anterior, and the posterior, which is the neural hypophysis. And you have 6 hormones produced by the anterior pituitary from 5 different types of tropic cells. One of them produces 2 different hormones. The gonadotropes produce follicle-stimulating hormone and luteinizing hormone. The other ones just produce 1 hormone. And then you have the posterior pituitary, and the posterior pituitary is going to produce 2 hormones, oxytocin and vasopressin. Another name for vasopressin is anti-diuretic hormone because it's anti-diuretic. Diuretics make you urinate. Anti-diuretic inhibits urination by reabsorbing the water from the nephrons. But anyway, so that's a lot of stuff from endocrinology you don't really need yet, but I'm just kind of like putting some of this stuff into perspective. You can see what this area of the brain has a lot of stuff there. That's why it's like the second brain, because it has so much of a regulatory type of control in your body. Okay. All right. So those are the 2 sections of the forebrain. Then you have the brain stem. So carry this down here. We'll go to the brain stem next. So this is the other major part of your brain, the brain stem. And the brain stem can also be divided into 2 parts, the mesencephalon and the rhombencephalon. So we'll go mesencephalon first. The mesencephalon is just simply your midbrain. There's a lot of structures on the midbrain, but just in general for now, we'll just call this the midbrain. And again, you can see we have the prefix mes, and then we have the root encephalon, which means brain, so midbrain. Okay, then we have the rhombencephalon. The rhombencephalon is your hindbrain. Okay, so here we have rhomb, which means hind, and then we have encephalon. Rhombencephalon means hindbrain. So again, you have rhomb, which is hind, and then encephalon, which is brain. And the rhombencephalon is subdivided into 2 parts. It's subdivided into the metencephalon. Metencephalon is your afterbrain. And that's going to basically be your pons and your cerebellum. Okay, and then the last part is going to be the myelencephalon, or myelencephalon. And the myelencephalon is your medulla oblongata. So you got met, then encephalon, afterbrain. Myel means spinal, so this is your spinal brain, which makes sense. Because the medulla oblongata becomes your spinal cord after it goes down through the foramen magnum. So hence we have spinal brain. And when you guys are out in practice, you'll see that you send out patients for MRIs, and sometimes from the reports you'll see you'll have compression of the spinal cord. The terminology for this might be like disc protrusion with myelopathy, right? Which just means pathology of the spinal cord. So you have abutment of the, you could have abutment of the herniated disc up against the dura, right? Or you could have actually compression of the spinal cord, which is a really bad kind of thing. And you got to be careful when you're adjusting those types of patients, because if there's herniated disc is pushing into their spinal cord and causing some deformation of it, you doing manual manipulative treatments could actually make your patient worse, or worst case scenario, even paralyze them. So, after being in practice for 34 years, I found, you have to address like disc herniations differently. So I like to use, Cox flexion distraction technique, and also spinal decompression, because then you're actually addressing the protrusion of the disc instead of just trying to manipulate the vertebrae, which is not going to really have an effect, because if you, by definition, the annulus, the nucleus pulposus, basically herniates from the annulus fibrosus. And adjusting is good because, it helps to keep things in line, but it doesn't do anything to bring that nucleus pulposus back in there. But if you suction it, traction causes suctioning, and it pulls that material back in there. And if you do that repeatedly, along with exercise and like a support belt, and the person doesn't have to do like any kind of manual labor, then I've seen pre and post MRIs, where the thing is significantly improved or has gone away entirely. So it's just something for you guys to start thinking about now. If you have the opportunity to take that, I think it's the elective here, that Cox flexion distraction, that's an excellent technique. Okay, so that's just my little tangent I went on with myelencephalon, myel being spinal. So myelencephalon is your medulla oblongata. Again, after that passes through the foramen magnum of the occiput, then it becomes your spinal cord. Okay, so this is a simplified version. I have a more complex version, but I'm just introducing you guys to some of the concepts here. Again, this is all going to, like the forebrain here, the proencephalon, and the rhombencephalon here, or the mesencephalon and the rhombencephalon are, you have the neural tube, right, which originates from the ectoderm, and then you get these outpocketings. And the first three outpocketings that develop into your brain are the forebrain here, or the proencephalon, the mesencephalon, and the rhombencephalon. And then those structures are going to, become all these structures that we put out here. And then the rest of it becomes your spinal cord. So that's just a little quick developmental kind of tie in there for you too. Okay, any questions on any of this stuff? So you guys know it already anyways. You went over it before with Dr. Decker, and now we're going to go into looking at some terminology, and then we're going to identify this terminology on some of these models here. Okay. So we're still working on structure. Remember we said we take each of our sections. I like to keep things super organized, and we do structures. Then we do muscles, nerves, arteries, and veins, where the topics are relevant. So we're on the structural component of this particular topic. Okay, so next, we're going to look at some terminology here. Still has to do with structure. In the terminology, we're going to look at grooves and like folds or elevations. So our first one is going to be a sulcus. Sulcus singular, sulci plural, is a shallow groove. And there's going to be two of these that I'm going to have you guys be aware of. the lateral sulcus, and, well, there's going to be three. The central sulcus, the lateral sulcus, and the parietal occipital sulcus. So, central sulcus. Lateral sulcus. The lateral sulcus is sometimes referred to as a fissure. And then we're going to have the parieto-occipital sulcus. And I'm kind of going down into slant. Okay. Then, so that's a shallow groove. That's the definition of a sulcus. Then we have fissures. Fissures are deep grooves. And the deep grooves that I want you guys to know is going to be longitudinal fissure. And the other one is going to be the transverse fissure. Okay, then we have notches. Notches are just indentations. And we're going to have two notches that I want you to know about. It's going to be a pre-occipital notch. Pre-occipital notch. And the posterior cerebellar notch. Okay, and then we're going to have start to go with, this is like the indented portions. Now we're going to go to the more, like the parts that protrude out. And we're going to have gyri, or gyrus, singular. Gyri is plural. And that's going to be an elevation, fold, or convolution, they're called. And there's going to be a few gyri that you should be aware of. We're going to have precentral gyrus. The precentral gyrus is called your motor cortex, because this is where you have the origin of all the motor signals that go to your muscles. Motor cortex. Then we're going to have the postcentral gyrus. Postcentral gyrus. And the postcentral gyrus is called the somatosensory cortex. Okay, so that's where we're going to have, all this incoming information after it goes to the thalamus. Then it's going to go to like the somatosensory cortex primarily, and then you have some association areas also in the parietal lobes of your brain. We'll talk about lobes of the brain here in a second also. Okay, and then maybe a couple other ones, because we have the limbic lobe of your brain that consists of the cingulate gyrus and the parahippocampal gyrus. And Dr. Main, in CNS, we'll talk about that. So let's include cingulate gyrus and the parahippocampal gyrus. Okay, and that's good enough. There's a lot of gyri there. Then we'll talk about lobules. And so a lobule is greater than a gyrus, but less than a lobe. That's my mathematical sentence there. Greater than a gyrus, but less than a lobe. And a lobe will be our last thing that we have here. And a lobe is a large structural slash functional region of the brain. Okay. And there's going to be 5 structural lobes, and there's going to be 1 functional lobe of the brain. Okay, so the lobules, I'm not going to hold you responsible for this quarter. There's like a precentral lobule. You're going to see that there's a supraparietal lobule. There's an infraparietal lobule of the brain. I'll let you guys have that in CNS next quarter, because we just want to introduce the main things here. And then but the lobes, you're going to need to know the 5 lobes of the brain. So there's 5 structural lobes of the brain. And those are easy. And so those are going to be 1, you have frontal lobe, 2, parietal, 3, temporal, 4, occipital, and 5. It's going to be insular lobe. The insular lobe is also called the Isle of Rail, like island. Maybe after the guy who found it or did a lot of research about it, so that's an R, rail. Let's see if I get confused with a P. Isle of Rail. And I'm going to point out all these different things that we've been talking about here in a second. Okay, so those are 5 structural lobes. Each of these lobes, they do have particular function associated with them, like the frontal lobe, complex, cognitive function, behavior, personality, all those kind of things in the frontal part there. The parietal lobes are going to be associated with sensory processing, like what we mentioned before, speech. The temporal lobes are going to be hearing. You have your major hearing cortex in that portion, auditory. Occipital is visual cortex, by a lot of visual kinds of things in the occipital one. And then the insular lobe has to do with intra perception of like your viscera, like the condition of your viscera, and also some of you as a person in general. So the insular lobe is kind of interesting that way. Okay, and then we're going to have 1 functional lobe. And the 1 functional lobe is going to be, that we're going to talk about, is going to be the limbic lobe. The limbic lobe is kind of cool. The limbic lobe associates your emotions to your learning and your memory. Right? So, like say you're on a weekend and you go take a drive somewhere and stuff, and you hear this song that you like, hadn't heard about it in years, and all of a sudden you get this emotional, welling up, and you think about stuff, and it brings back memories, right? So this is what the limbic lobe does. It associates your emotions to your learning and your memory. So it's a tie-in to your memory emotionally. So that's what it does. It associates emotion to learning and memory. Okay, and that's another reason why we'll talk even some of this stuff in a minute. That it's nice to have a positive environment for your classes and stuff, where you don't feel overly stressed, and you actually can have fun, because putting a positive emotional component to this process of having to study and learn will actually facilitate your memory and your learning. So you tend to remember things a lot better, like if you're enjoying them, right? That, along with organization and making it relevant to you as a person, those are things that help you to keep your memories. You notice the things that had bad happen to you, that usually people will tend to get rid of those as they go along. So, it's not a good, healthy thing to store negative kind of memories. You know, you have them in there, you know, for learning purposes, so that you know how to cope with them later on and stuff like this. But generally, you know, you tend to forget these things, and unless something causes you to bring them up, right? And then you basically remember the positive kinds of things. So that's part of the memory and the learning kind of thing. Okay, so that's those major things here, and we'll point this stuff out, and then maybe I'll do some cranial nerve stuff, and then we'll have done a lot of stuff. And the cranial nerve stuff is important. You know, most of the cranial nerves are going to emerge from the brainstem. You know, we just talked about that structural organization of the brain, and they all have specific function, and we're going to really hit those. This section, the intracranial section, I want you guys to know where they are. Right, I have one of the content support drawings for this time around is going to be a picture of the ventral brainstem, which you're going to see again when you go to CNS, and you need to know where these cranial nerves are in association with the ventral brainstem. And then we're going to talk about the major functions of them in this particular section. Next section that we do extracranial at the end of the quarter here, our last section, most of the stuff extracranial that we talk about is going to be innervated by these cranial nerves. So we're really going to get into cranial nerves at that point. So I'm going to kind of set you up for them with some of the basics. Then we're going to get more specific in there. The only thing that I'm not going to teach you about the cranial nerves this quarter is going to be their functional classifications. But I have a real easy way to do the functional classifications, and you will need to know those for next quarter. So if you're interested in that, then I could send you a little outline that I made, how to construct these, you know, the functional classifications, so you don't have to memorize it. Because if you try to memorize this stuff, you'll see that there's a lot of stuff there. But the way I do things, I don't like memorization. So I will show you functionally how you can reproduce this in a very simplified manner, and then you'll actually understand where it's all coming from. In the pictures, you know, like, and it's based on development also, and it's only like one page. Okay? So if you're interested in that, just remind me. All right, let me get a pen here. We'll take this pen, and then let's start to look at some of these things. You guys can come up. We'll find out some structures, you know, lab.
Thank you, Ray.
Okay, so, you know, just so you can kind of get a, you know, feel for some of this stuff, you know. For when you're first learning the brain, this one is nicer because it's not as convoluted. You don't have as many folds. Like this one here, you have a lot of convolutions, right? So it might be good to start out learning this particular one. In fact, I could probably do a little bit of a drawing also too, and we can label. I have content support drawings for this stuff too. But then after you kind of master this one, then you can go to a more convoluted brain like this type. And by the way, you know, it was you know, theorized that people that have a lot of convolutions in their brain shows greater intelligence, although some of that research, you know, some scientists are trying to refute. But you know, like a lot of research through in Russia and stuff will show, you know, that's more current, that you know, that where there is convolutions, especially in like the pathways between like your visual and your complex problem-solving areas in the frontal lobe, there is a sign of higher intelligence. And we'll talk about it also too, like with Albert Einstein's brain. You know, Friday, I have, you know, a picture on the PowerPoint of Albert Einstein's brain. It was taken out of his skull. You know, it's like 7.5 hours after he died. It wasn't even authorized, but they just went ahead and did this. And they actually also took his eyeballs out, and they gave it to his eye doctor. You know, so they basically, he flipped out people so much with his intelligence that, and people just didn't understand him so much that they were just, they wanted to know what made him different from everybody else. That's why they did all this stuff. Okay, and then so we'll talk. I'll save that for you. So Friday we'll talk more about you know, like basically what they found there. Okay. Anyway, so here is like the brain, looking at it laterally, and then so we'll look for things that we have here as far as terminology goes. So sulci, we said, are shallow grooves. The sulci that I want you to know is the central sulcus, So the central sulcus is this one here. It goes all the way down from the fissure of the brain here. This is called the longitudinal fissure. It's one of our things too for fissures. But, and then it goes down to this one, which is the lateral sulcus. So you have the central sulcus. We have the central sulcus. It goes all the way down uninterrupted, pretty much. And then we have the lateral sulcus, which is this one, right? So the brain kind of looks like maybe a mitt or a punching, a boxing glove or something, right? And so the lateral sulcus is going to separate the frontal and parietal lobes from the temporal lobe here. The thumb portion would be the temporal lobe. So central sulcus, lateral sulcus, and then the other big one was the parietal occipital sulcus. So parietal occipital sulcus, separating the parietal lobe from the occipital lobe. So this would be the parietal occipital sulcus here. Okay, everything behind the parietal occipital sulcus is going to be occipital lobe, and we said that this is the center for visual processing. In fact, you're going to also learn this sulcus here is the calcarine sulcus, and the calcarine sulcus has a lot to do with, that's like the center of the cortex around the visual processing. Oh, and when I was talking to you guys about those convolutions and that the reason why the brain convolutes is because to increase the surface area of the brain in a non-expanding volumetric enclosure, like your skull is, once it fuses, right, it doesn't expand. So in order for your brain to have greater surface area, it has to kind of fold in on itself, right? So that's how you get those convolutions. Okay, so central sulcus, lateral sulcus, parietal occipital sulcus. So this parietal occipital sulcus is kind of funny because it only comes up and it goes to about here, then it ends. Okay, so we have to kind of draw it in there to kind of separate on this side in order to separate the other parts from the occipital lobe. And this is where we're going to use this other notch. If you look on here, we have the pre-occipital notch. The pre-occipital notch is like the highest point above the cerebellum. So you take this parietal occipital sulcus up to where it ends, find the pre-occipital notch, again, the highest point above the cerebellum, and then you make it like a dotted line connecting the two. So like this kind of a plane line. So everything behind the pen, therefore, is going to be occipital lobe. So that's how basically scientists do this, you know, in order to kind of know, you know, the area of the brain as far as like the lobes go. Okay, so those were our sulci: central sulcus, lateral sulcus, parietal occipital sulcus, and I showed you how to draw it. Okay, then we have fissures. We have two major fissures. We have the longitudinal fissure, which is this one, separating the cerebral hemispheres. Then we have the transverse fissure, which separates the cerebral hemispheres from the cerebellum. So here's your longitudinal fissures here, right? And then between your cerebellum and your cerebral hemispheres here, this would all be transverse fissure. Okay? These things are important because you're going to see, well, you probably talked about it already with Dr. Decker because you guys went over the meninges. There's going to be areas of the brain where the meningeal layer of dura mater is going to infold into some of these crevices, right? Did you talk about like the falx cerebri? Mm-hmm. Yeah, so like the examples of the falx cerebri or the falx cerebelli or the tentorium cerebelli or the diaphragm ocelli. These are all areas where the meninges will infold into these crevices. Whenever this happens in the brain, this allows for the presence of those durovenous sinuses also. So those are like the distended veins. They would be located in these infolds of these meninges, in these crevices. Okay, so that's why you guys have to be familiar with where they are. So longitudinal fissure and transverse fissure, since you already had that, the longitudinal fissure is going to have the falx cerebri, which is going to contain the superior sagittal sinus and the inferior sagittal sinus. Inferior sagittal sinus will meet up with the great cerebral vein of Galen to form the straight sinus. Okay. The transverse sinus basically has two different types of durovenous sinuses. The transverse fissure contains the falx cerebelli, it contains the tentorium cerebelli. This transverse fissure contains the tentorium cerebelli, and tentorium means tent, right? So it looks like a tent-like structure. The tent has a roof. The roof of the tent is going to be the straight sinus, and the floor of the tent on both sides is going to represent the transverse durovenous sinuses. I can tell you guys this because you already had that. You guys are just learning this stuff for the first time. You don't need to know that yet. Okay, so those are our fissures. Some people call this lateral sulcus also the lateral fissure or the Sylvian fissure. That's just another thing that some of them do. Okay, and then we had a couple of notches, right? We have the pre-occipital notch, I already talked about that. And we have the posterior cerebellar notch here between the cerebellar hemispheres. Now the posterior cerebellar notch is going to contain the falx cerebelli, and the falx cerebelli is going to contain the occipital sinus. Okay. All right, so those are all of our grooves. We did sulci, fissures, and notches. Then we talk about the gyri. We're starting to go into the elevated portions. So the gyri, we said, are elevations, folds, or convolutions, and there's a few of them that I want you to know. The first one is in front of the central sulcus. So the gyri are usually named according to the sulci, to which they are closest. So this is the central sulcus. So this fold here in front of the central sulcus is called the precentral gyrus, right? So that's the precentral gyrus, also known as your motor cortex. So this is where you have you have the motor signals to all of your muscles is going to originate. Okay, and then behind the central sulcus, we're going to have the post-central gyrus, which is also known as the somatosensory cortex, because this is where we're going to have afferent information from all sensories coming into the thalamus, and the thalamus is going to direct a lot of those, the somatic portions of them. And you know the difference between somatic and visceral, right? Somatic is going to be your muscles, your tendons, your joints, right? Your viscera is going to be your organs, right? So this is somatosensory cortex processing. So that's where this information is going to go sensory-wise to be processed. Okay, and then we had a couple of the gyri that I mentioned. The cingulate gyrus is this one here. This is the cingulate gyrus, the part of the limbic system. And then we had the parahippocampal gyrus. The parahippocampal gyrus kind of surrounds the brain stem. So if you take off the cerebellum, then surrounding the brain stem, this area here would be the parahippocampal gyri. And why are they parahippocampal gyri? Because yeah, and the hippocampus, you guys know this, and the hippocampus is what? It's your primary learning area, right? This is where most of your learning takes place, is in the hippocampus. So if you take the brain apart like this, right? Don't try this at home. And usually we'll have the corpus callosum, which is going to overlie that, but the little piece is missing. The corpus callosum is a nice little kind of reference because it represents the largest aggregation of white matter in the brain. And the white matter in the brain is myelinated axons. And a type of myelinated axons are commissural fibers which commute between the right and left halves of the brain. This is also the corpus callosum here. You can see on this particular view. So it would be sitting over the top of that, where this area here is the corpus callosum. Okay, and then you continue to take all this stuff off. So we're wanting to know about the hippocampus. So we take out the brain stem, right? And then these two green lobes are your hippocampus of your brain. So that's where most of the learning activity takes place there. On the other side of them is that parahippocampal gyrus. And then we said that the limbic lobe is a functional lobe, and it associates your emotions to your learning and memory. And I forgot to put down that the limbic lobe actually consists of the cingulate gyrus plus the parahippocampal gyrus. cingulate gyrus plus the parahippocampal gyrus forms the limbic lobe. Okay, so here's your cingulate gyrus up above this corpus callosum. This gyrus, along with the parahippocampal gyrus, makes up the limbic lobe of your brain, which is a functional lobe, tying your emotions to your learning and memory. There's a lot of other limbic structures, like you'll see you have the amygdala, which is the emotional center, basal nuclei of the brain, is one of them. You have this fornix, you have these mammillary bodies. These are all extra limbic types of structures that have to do with emotions. And you have like reward centers that are actually tied in with those emotional centers, like the nucleus accumbens. Those are all basal nuclei or ganglion. So remember we said, you know, you can take, well, when we did the structural organization of the brain, you had the proencephalon, which consisted of the telencephalon and the diencephalon. The telencephalon were the cerebral hemispheres. The cerebral hemispheres have an outer white cortex, centrum semi-ovale, which consists of the white matter, different tracts. And then you had these basal nuclei at the base of the brain. Some of those basal nuclei are like the amygdala for the emotions. You have the lenticular nucleus, which is like the globus pallidus, and the putamen. And you have the caudate nucleus, which has to do with complex cognitive functioning. Okay? Those are all examples of basal nuclei. Okay, so let's see, we did that. Talking about those things. Lobules, I said lobules, you don't, I just got done talking about gyri. Lobules, we're not going to really get into lobules too much. Lobules you're going to have, and I'm not holding you responsible for this, but we have this sulcus here, which looks like, you ever see people that do a simplified bird picture, right? So, you know, like when they make a bird on a picture, they go like this, you know? So it kind of looks like that. That's the intraparietal sulcus there that looks like that. Above there we have the superior parietal lobule, and below that we have the inferior parietal lobule. Okay, and in the area around here, it's called the supramarginal gyrus. You don't need to know that yet, but that's where you have that Wernicke's area for language. Okay, and let's see what else. Okay, so and then we have another lobule in this area here called the paracentral lobule. So this around where the central sulcus comes through and comes on this side, around it is the paracentral lobule. But you don't need to know that stuff. You will need to know though for CNS next quarter. I'm just sticking to the basics. Okay, and then we move into the lobes. So we have the frontal lobe, which is everything in front of the central sulcus, is the frontal lobe. Behind it is going to be the parietal lobe. Okay, so the precentral gyrus is in what lobe?
Frontal lobe.
It's in the frontal lobe. And the somatosensory gyrus is in what lobe? Parietal lobe. Okay, then we get to our designation for the parietal occipital sulcus, and then everything behind that will be occipital lobe. This is the temporal lobe here. Looks like the thumb part to a boxing glove. Okay, and then the insular lobe is actually here. And so like if you put this thing back together, the insular, I like to use this as a test question on my labs. I don't know how Dr. Decker does. Everybody has their own thing, you know. But if you put this brain together like this, the insular lobe, you know, I might have a question like this, and this popped up like that with a tag on this thing. So then you would know that's the insular lobe of the brain. So that has to do with interoception, so it gives you information about, like, you know, the integrity or what's going on with your internal organs. Okay. All right, so those are all those things. Let's take a break, and then we'll do cranial nerves real quick, and then we'll call it for today. You get out of here somewhat early.